And what is the quantum chemistry? The quantum chemistry, or more precisely molecular quantum mechanics, is the application of principles of quantum mechanics to the molecular systems. The first step of solving quantum chemical problem is solving of Schrödinger equation with the electronic molecular Hamiltonian. An exact solution, as it is very well known, can be only obtained for the hydrogen atom. For other systems which consist from more than two particles, exact solution of Schrödinger equation is impossible. And this is the reason why we need to choose some approximation to perform a numerical solution of Schrödinger equation. And the zoo of the common quantum chemical approaches or methods can be visualized as the so-called Jacob's ladder, where they are ordered from the Hartree wall to the heaven of chemical accuracy. And after choosing of appropriate approach, we perform solving of Schrödinger equation using some computer resources. Depending on the size of molecular system, chosen approach and available computational resources, this solution can take from parts of second up to months or maybe even years. And after that, when we solve Schrödinger equation, we obtain electronic structure of molecule. And electronic structure of molecule is responsible for any chemical properties of molecular substances. And in our lab, we developed a number of projects, including quantum chemistry methods. And all projects can be classified into two categories. The first category is devoted to investigations of spectral parameters, or more precisely finding a correlation between spectral parameters such as IR or NMR spectral parameters with some properties of non-covalent interactions, such as their energy and geometry. And this direction is quite interesting because spectral parameters sometimes are not quite easily obtained from experiment, and computational data can be used for either interpretation of experimental data or even for modeling of impossible experiments. And the second direction is devoted to visualization of features of outer electronic shells of molecular systems in order to predict their reactivity and also for calculating some thermodynamic parameters of various chemical reactions. And the topic of my today talk is actually related to both directions, and it's a quite new project for us. And all of you, I am on the first listeners of results of our project devoted to investigation of selenium-containing enzyme glutathione peroxidase. Selenium is a chemical element with atomic number 34. It's a metalloid whose chemical properties is somewhere between their neighbor above and below in chemical periodic table, sulfur and fluorine. Selenium has 6 stable isotopes, with selenium-80 being the most abundant one. From the NMR point of view, selenium-77 isotope is the most interesting one, because it has spin 1/2, relatively high natural abundance, and quite a broad range of chemical shifts. In ambient conditions, selenium exists in the red form, but with increasing of the temperature, it converts to the black form, which looks like a lustrous black solid with irregular structure, consists from polymeric fragments of selenium atoms. In biological systems, selenium found as the residues of 2 selenium-containing amino acids, selenocysteine and selenomethionine. These 2 acids differ from their sulfur analogues, cysteine and methionine, only by replacing of only one atom, sulfur by selenium. But the chemical properties of sulfur-based and selenium-based amino acids are completely different. For example, selenocysteine is more than 3 orders of magnitude stronger acid as a cysteine. Selenocysteine also has much higher nucleophilicity than cysteine. And probably this difference between sulfur and selenium-based amino acid is caused by some special properties of selenium atom. Maybe it's higher polarizability or an isotropic distribution of electronic density near selenium atom. Selenocysteine in human cells usually found in selenoproteins. Selenoproteins is proteins which contain seleноcysteine amino acid residue. And at the moment, there are 25 identified selenoproteins in human cells. They can be classified into several families. The first family is iodothyronine dionidases. This family is responsible for the activation and deactivation of thyroid hormones. There are also 3 thyroidexine reductases, which controls level of thyroidexine hormone. And another large family is glutathione peroxidases, which are responsible for the protection of human cells against oxidative stress. And in this work, we focused on one member of glutathione peroxidases family, glutathione peroxidase 1. Now it is well known that the function of this enzyme is associated with antioxidant activity. However, the mechanism of its action remained largely unknown. And the possible mechanism of catalytic action of glutathione peroxidase 1 is shown on slide. At the first stage, selenol form of amino acid is deprotonated. After that, it interacts with hydroperoxide molecules, form the single oxidized form selenium OH, which subsequently reacts with the glutathione molecule as a cofactor. And after that, selenium sulfide form is formed. The subsequent nucleophilic attack of another glutathione molecule is accompanied by a removal of the oxygenated glutathione form and recovery of initial enzyme. At higher concentration of hydroperoxide in the systems, additional step is possible. Selenium OH form may undergo further oxidation with the corresponding selenic acid formation, and which will also converted to selenyl sulfide form by reacting with an excess of glutathione molecules. At all stages of this possible catalytic cycle, glutathione peroxidase active center can be involved in various non-covalent interactions with neighbor amino acid residues and molecules of cofactors. However, at the moment, it is not proved that this catalytic cycle is the real catalytic cycle, and it's also nobody knows which intermediates form at each step of this catalytic cycle. The answer to these questions can be important for the development of future antioxidant selenium-based drugs, or for some purposes of organoselenium chemistry. And so on this work, we decided to answer some of these questions. So all of these projects are as follows. The first goal is investigations of features of outer electronic shell of model systems, which can be considered as a mimics of selenosystine moieties in a vacuum. After that, we investigate the hydrogen bonding with the water molecules for the model systems. Then we investigate non-covalent interaction that exists in the real enzyme, and then compare results obtained for a simplified system and for real systems. And as I mentioned above, we use a quantum chemistry as the main method for our investigation. And so let's move on to the results. On this slide presented are the optimized geometries of selenosystine moieties along the catalytic pathway, its initial form, deprotonated form, oxidized form, double oxidized form, and selenium sulfide form. And on the bottom part of the slide presented are the distribution of electrostatic potential for all of these compounds. And it is clearly seen that features of outer electronic shell of selenium-containing fragment changes significantly during the catalytic transformation. For example, the negative charge for deprotonated form B is localized in toroidal-shaped region around selenium atom, perpendicular to carbon-selenium bond. On the continuation of carbon-selenium bond, there is a region of maximum electrostatic potential, which can be a potential spot for potential nucleophilic attack of peroxide molecule. For the oxidized form C and D, it can be seen that OH protons are quite acidic. Such significant changes in electronic shell can be also visualized by using other techniques. But what about the thermodynamic parameters of catalytic cycle based on such simplified system? And it's clearly seen that deprotonation of initial selenium form is accompanied by the energetic gain of about 15 kilocalories per mole. The further oxidation is also exothermic processes with the release of about 16 kilocalories for 2 steps. And this result shown that the presented reaction is energetically favorable, and it's also an argument that the proposed mechanism can be a real mechanism. And such dramatic changes in thermodynamic parameters and electronic shell features can be also tracked by using selenium-77 NMR. It's clearly seen that calculated chemical shift of selenium atom for forms of selenium containing moieties changes on about on more than 1000 ppm. Initial form of enzyme in its deprotonated form has selenium chemical shift of about plus 100 ppm. Oxidized and double oxidized form has signal in the range of about minus 1100 ppm. And selenium sulfate form has selenium chemical shift of about minus 400 ppm. And thus, it can be shown that the measurement or calculation of selenium chemical shift can be a source of reliable information about the degree of oxidation of selenium-containing fragment. As we discussed above, water molecules are products of reaction of selenium-containing fragments with peroxide molecules. And thus, these water molecules can participate in the formation of hydrogen bonds with selenocysteine residue. And the directions from which water molecules can approach selenium-containing fragments are restricted by the anisotropy of outer electronic shell of selenium. And it could be illustrated that topological features of electronic shells can be visualized using so-called electron localization function, whose maxima usually demonstrate the electron-rich region, such as lone pairs or something like that. For initial selenium form, it can be clearly seen that there are 2 regions of ELF maxima, which is responsible for 2 spots for coordination of 2 water molecules. For deprotonated form, there is the toroidal shape region around selenium atom, and these spots are also can be used for water coordination, and their numbers is limited only by steric reasons. For oxidized form, there is also 2 spots for coordinations of water molecule. And this maxima located perpendicularly to carbon-selenium hydrogen bond at approximately 120 degrees from selenium H bond. And this localization attractors determine that only 2 water molecules can be accommodated in the first coordination sphere of such selenol moiety. And in order to understand how the hydrogen bonding with the water molecule influence the electronic shells and some spectral parameters of such moieties, we perform an additional investigation. We saturate coordination sphere of initial enzyme, deprotonated form, and oxidized form by water molecule, and optimize geometry of all of these structures. And we found that 2 water molecules are can be accompanied in the first coordination sphere of initial selenium fragment, as it was predicted from the ELF distributions. 6 water molecule can be located near deprotonated form, and 2 water molecule can be coordinated to selenium atom in selenium OH form. In order to visualize the electronic shells feature changes upon the saturation of first coordination sphere of selenium by water molecules, we perform the sequential addition of 1, 2, 3, 4, 5, 6 water molecules in the coordination sphere of each of these species. And on this slide, I show structure of complexes with deprotonated form, but other forms are also investigated. But for the sake of simplicity and clarity, I will discuss later only the behavior of deprotonated form. And we found that the electronic distributions near selenium atom are significantly changes upon the formation of hydrogen bonds with water molecules. For example, the deprotonated form without any connection with hydrogen, with water molecules has electrostatic potential shown in the left part of the slide. On the middle part of the slide, you can see deprotonated form bonded with 3 water molecules. And on the right part of the slide, you can see deprotonated form of seleноcysteine fragment connected with 6 water molecules. And it's clearly seen that electronic distribution changes significantly. For example, on the continuation of carbon-selenium bond for isolated species, electrostatic potential reach of about minus 0.2 atomic units. A formation of hydrogen bonds with 3 water molecules leads to increasing of maximum of electrostatic potential value on the continuation of carbon-selenium bond on about 0.1 atomic unit, and for fully saturated coordination sphere with 6 water molecules, the maximal value of electrostatic potential reach of about 0 atomic units. And this plot, as I mentioned above, can be used for coordination of hydroperoxide molecule. And so on this example, we can conclude that the formation of additional hydrogen bonds promote the support for further nucleophilic attack of hydroperoxide. These significant changes in the electronic shell can be also tracked by using of selenium-77 NMR. And from this slide, it's clearly seen that computed chemical shift of selenium atom approaches the experimental value only for almost fully or fully saturated first coordination sphere of selenium. So we can speak that saturation of first coordination sphere of selenium is essential for the accurate prediction of experimentally observed values. And my talk to this moment was about simplified systems that can be used as very simple models of seleноcysteine fragment. And we understand that formation of even weak hydrogen bonds with water molecules cause significant changes in spectral parameters and electronic shell distribution. But what happened in the real enzyme? Which kind of interactions we can found in the active site of enzyme? The real enzyme is tetrameric. It has a X-ray structure shown on the left part of the slide. This structure is quite large. It contains more than 6,000 atoms, and working with such a large system by means of quantum chemistry is almost impossible. One step of optimization for structure like this can take for about years. And this is the reason why we decided to somewhat simplify our model system. We cut the active center from one fragment, and we found that the active center looks like this. Balls and sticks are used for visualize seleноcysteine moiety, and the tubes visualized another amino acid residues, and 2 water molecules are also found in the active center of the enzyme. These 2 water molecules exist in the active center due to the seleноcysteine moiety is in double oxidized form. Thus, the reduction of 2 peroxide molecules was happening earlier. And we worked with systems containing of 88 atoms. It's also quite complicated for quantum chemistry, but we spend a lot of days, even months, trying to optimize the structure and working with thermodynamic parameters. And what we found it. The optimized structure of these complexes shown on this slide. It's clearly seen that Selenocysteine moiety is involved in many non-covalent interactions, or some short contacts. But in order to classify these short contacts, perform quantitative investigation of the strengths of these contacts, we perform additional step. We perform topological analysis of electron density distribution using Bader's theory of quantum theory of atoms and molecules. And we found that selenocysteine moiety is involved in many non-covalent interactions, in 1, 2, 3, 4, 5, 6, 7 hydrogen bond and 5 other interactions. Let's start from the hydrogen bonds. There are 7 hydrogen bonds. The strongest hydrogen bond is formed between selenium OH group and one of water molecules. Its strength of about 40 kilocalories per mole. Another 3 strength hydrogen bonds are formed by selenium double bond O with oxygen molecule and with a tyrosine residue, its strength of about 10 or 11 kilocalories per mole. And also selenium O group is also involved in quite strong hydrogen bonding with an H group of glycine. Its strength is also about 10 kilocalories per mole. Another 3 hydrogen bonds are much weaker. Its strength lies in a range from 1 to 4 kilocalories per mole, but these hydrogen bonds are also important for the formation of the pocket in the active site inside of enzyme. And what about another interactions? Another interaction will be categorized using the electrophile nucleophile criterion, which is proposed in later work of Bardashevich. And we found that 4 of these short contacts are chalcogen bonds, and the last one is the pnictogen bonds. The strength of all of these interactions is less than 6 kilocalories per mole, but all of these are important. The strongest contact is the chalcogen bond formed between selenium atom and the oxygen atom of asparagine amino acid residue. Its strength of about 6 kilocalories per mole. All other chalcogen bonds and 1 pnictogen bond has energy less than 2 kilocalories per mole, but they are also take a significant impact on what happened in the active site of the enzyme. And let's compare what happened in the real enzyme and calculate thermodynamic parameters of catalytic transformation along the proposed catalytic mechanism for this real system, and compare it with previously discussed result for the simplified model system. For example, on the left part of the slide, you can see one step of catalytic transformation for simplified mechanism based on selenocysteine molecule with formation from single oxidized form of double oxidized form. This process in vacuum for selenocysteine is accompanied by the energetic gain of about 50 kilocalories per mole, or maybe some less. And for a real enzyme, for the active center of enzyme, more precisely, this process is accompanied by a more significant energetic change. The energetic gain is about minus 55 kilocalories per mole. And the difference between the vacuum case and a real enzyme is about 6 kilocalories per mole. From the practical point of view, it's quite small number of kilocalories per mole, but we should take in mind that it's only about one step of catalytic mechanism. But energetic gain on another steps is also quite large. It takes more 5 to 10 kilocalories per mole. And so the energetic gain for the whole cycle can reach for about 15 kilocalories per mole. So this weak interactions that we take into account modeling quite complicated systems with participation of various non-covalent interaction promote the formation of double oxidized form and stabilized its structure. And so in the conclusion of this part, I would thank the organizers for the opportunity to take this talk today. And I can conclude that the result of our project can be used for further development of modern antioxidant drugs. And we show that taking into account of even weak non-covalent interactions formed inside the real enzyme are quite important to accurate prediction of thermodynamic characteristic of glutathione peroxidases enzyme catalytic cycle. And electronic shells of selenium atom is really able to be easily polarized, and this changes in the electronic shell can be easily tracked by means of selenium-77 NMR. And this project is only one project among the many other we realized in our laboratory, and students that work in our group participated in many research projects at the same time. And if you are interested in what we are doing in our laboratory, you can also listen speeches of other members of our laboratory. For example, yesterday, Mr. Valery Karpov, our master student, talked about the computational protocol of correct calculation of spin-spin coupling constant between carbon and lithium. Today, in this section, two talks will be presented from members of our group. One by Demchikov about experimental investigations of quite strong or hydrogen bonds formed by phosphinic acid and protonic sponge. And another talk from member of our group is by Anna Titova about the mutual influence of three coupled NHN hydrogen bonds. And the other talk will be presented in Thursday by Mark Kaplansky about theoretical investigations of proton chemical shift for central symmetric systems. And the last talk by Mr. Vladislav Korostilov about the visualization of aromaticity and anti-aromaticity for cyclic compounds using helium-3 chemical shift as a probe. And if you have any questions, I will be glad to answer.
Thank you for your presentation, for your lecture. And please